Hello students, welcome to EPG Pathshala. I am Vidya Kothekar and my introduction is for the last 50 years I have been a teacher at All India Institute of Medical Sciences, JP Institute of Information Technology Noida, Dr. D.Y. Patil Biotechnology Bioinformatics Institute and so on. And I love teaching and teaching students in different subjects. Today, I am going to discuss about a module, Chargaff rules and DNA polymorphism. These two topics come under the paper Biomolecules and their interaction. DNA molecule under different environmental conditions and with the non-random sequence adopts different overall structures. It also shows short range and long range structural changes. These features make it a very suitable molecule which not only carries our genetic information but is also amenable to the environment and environmental attacks, and it modifies itself and corrects itself in the different situations. Students, let us first see what we are going to learn in this module. So first, I want to discuss the historic Chargaff's rule concerning base composition in different organisms. It is important because DNA adopts different structures known as polymorphic forms of DNA if the base composition changes or environment changes and many other things change. Then we want to elaborate on environment and sequence dependent conformational changes in DNA. I wish to introduce you to the DNA multiplex structures. Chargaff's rules. What were these rules? These rules are called Chargaff's parity rules. As early in 1952, an Austrian chemist, Elson Chargaff, proposed a rule that DNA of any cell organism should have 1:1 ratio of pyrimidines and purines. The first parity rule states that double-stranded DNA has percentage of base equality. That means percent %A equal to percent %T and percent %G equal to percent %C. The rigorous validation of the rule constitute the basis of DNA structure given by Watson and Crick. Chargaff's rules. The second parity rule states that percentage of A is equal to percentage of T and percentage of G equal to percentage of C. These are valid for each of the two DNA strands. This describes only the global feature of the base pair composition of the single-stranded DNA. The Chargaff's rule also states that composition of DNA varies from species to species. In particular, the amount of A, G, T, C. Such evidence makes it a better candidate for genetic material. We show in this table the composition of nucleobases in different organisms. For example, in case of maize, percentage of A is 26.8% and percentage of G is 22.8%. Percent of C is 23.2%, which is very close to the percent of G. And similarly, percent of T, which is 27.2% and very close to percent of A. Thus, the rule seems to be valid. Another example we can read is, say, chicken. A is 28% and T is 28.4%. G is 22% and C is 21.6%. E. coli. E. coli has 24.7% A and 23.6% T and 26% of G and 25.7% of C. We can also compare the percent GC with percent AT. Some of the organisms are richer in GC sequences and some of the organisms are richer in AT sequences. Just take the case of human. Percent AT is very high compared to percent GC. AT is 59.6%, whereas GC is 40.7%. Similarly, wheat, the percent of AT is 54% and GC is 45.5%. The case is reverse in case of E. coli. GC is 51.7% and AT is 48.3%. Chargaff rules became very important. In the year 2006, it was shown that only a few types of double-stranded genomes, example, eukaryotic chromosome, the bacterial chromosome, the viral chromosome, and archaeal chromosomes, the rule is valid. It does not apply to organellar genomes such as mitochondrial or plasmid or plasticid genome smaller than 30 kbp, nor does it apply to single-stranded viral DNA or any type of RNA. Polymorphic forms of DNA. The B form of DNA is realized for a random nucleotide sequence in a highly hydrated state. In lesser hydration, say less than 30%, DNA assumes A form, which is more crystalline in nature. The C form of DNA was found in lithium salt at low relative humidity and in the presence of sodium salt in high salt and humidity condition between A and B DNA. We show in this picture A and B forms of DNA. The pictures are drawn using Rasmol, and what we can see is the B form is a very curvature based form. It's very pretty, long and slim, whereas A form is more fatty or stout and short form. Similarly, the bases in case of B form are perpendicular to the helix axis, whereas in case of A form, these are inclined compared to the helix axis. You can notice it from the picture. C form of DNA has axial rise per residue 3.31 angstrom. It has pitch 33.7 angstrom and rotation per residue 38 degrees. Similar to A and B forms, it had a right-handed helical structure. cDNA has a minor groove width 4.8 angstrom deep and depth 7.9 angstrom. Major groove width is 10.5 angstrom and its depth is 7.5 angstrom. Physicochemical studies on DNA. During mid-60s and 70s, number of studies were carried out on natural and synthetic DNA fibers, also on oligonucleotides under different environmental conditions. The techniques used were fiber X-ray diffraction, single crystal X-ray diffraction, differential thermal analysis or DTA, nuclear magnetic resonance, CD spectroscopy, ORD spectroscopy, UV spectroscopy, fluorescence spectroscopy, etc. Plethora of DNA conformations became available through these studies. Physicochemical studies showed that DNA has a tremendous capacity of adopting plethora of conformations. It can have C, C dash, C2 dash, D, E, and so on. Some of these structures differed marginally, while others had major differences. These conformations were named using single or multiple alphabets. As a matter of fact, all alphabets except F, Q, U, V, and Y have been used to designate DNA conformations. Left-handed DNA structure. A dramatically different structure was seen in a hexanucleotide duplex with the sequence CG, CG, CG. This structure was solved by Wang and Alexander Risch and many other authors. This was named as Z-DNA. It had a unique left-handed structures. All alternate pyrimidine-purine sequences except alternate TA showed this typical unique left-handed structure. Structural features of Z-DNA or left-handed DNA. G-DNA has a dinucleotide repeat. All other DNAs have mononucleotide repeats. Then purines are towards periphery. Like in B-DNA, purine, pyrimidine are near the center. Sugar pucker with purines is C3' endo. But pyrimidines have C2' endo sugar conformation. Purines have syn conformation of the bases, while pyrimidines have anti-conformation. The average width of this DNA form was 18 angstrom. It had 12 base pairs per turn, giving rise 48 angstrom as pitch. We show in the picture space filling model of left-handed DNA drawn with using 5-mole software. cDNA has practically no major groove. The width is 2.0 angstrom and depth is 3.7 angstrom. It has deep 13.8 angstrom and narrow, it's not narrow, little wider, 8.3 angstrom, minor groove passing through the center of the structure. We show in this picture a typical characteristics of left-handed DNA. We can see the guanine shown as red here and cytosine are shown with the CPK colors, that is grey color. And the sugar with the cytosine stacks on guanine. The sugar-based stacking is a very, very unique property of left-handed DNA structure. Another right-hand side picture, we show only the backbone trace of a dinucleotide. And you can see it makes like a letter of Z rather. That is why it is called left-handed DNA structure. In this picture, we show comparison of A, B and G DNA. All these three structures have 12 base pairs and you can see the difference in the relative heights, like G DNA is tall, B DNA is medium with the curved grooves, A DNA is a short and stout. The structures are drawn with the 5-mole software. In the gDNA, the backbone goes, shown in the case of red color, that goes to the left direction. In bDNA, it goes in the right direction. So also aDNA, the blue backbones in A and B form are going towards the right hand direction. Suppose you look a 4 base pair sequence of DNA, A form, B form and G form in the view perpendicular to the helix axis, we can call it top view. Now, in case of bDNA, you can see all the bases clustered near the helix axis. In case of aDNA, the bases are pulled away from the helix axis. This actually makes a hole. That is, if you see from the top, you see a hole in the center of the structure. In the zDNA, it is a mixed picture. The guanine is towards the periphery, whereas cytosine is towards the center of the and closer to the helical axis. Comparison of structural parameters of A, B and cDNA. We show in this table various structural parameters of B form, A form and G form. As you can see immediately, that helix sense in the case of B and A form is right-handed, whereas in G form it is left-handed. Pitch is equal to 3.38 angstrom in bDNA, 2.5 to 3 in aDNA, and gDNA it is 3.5 and 3.9 alternate. Twist angle, helical twist as we call it, it's 36 degrees in B form, 30 to 33 degrees in A form, and G form is a dimer, so we give average 30 degrees. Tilt angle, that is the angle between the base pairs and the plane perpendicular to the helix axis. It is practically 0, it is 1 degree in B form and the bases are almost perpendicular to the helix axis. In A form, it is 19 degrees bases are quite tilted with respect to the helix axis, whereas Z form, it is in between, it is 9 degrees, -9. We can compare the sizes of major grooves and minor grooves. As discussed earlier, in case of B form, it has a wide major groove, 11.7 angstrom, and deep 8.5 angstrom. And Z form has practically no groove. It is width is 2 angstrom and depth is 3.7 angstrom. A form, it's a narrow deep major groove, 2.7 angstrom width, 13.5 angstrom depth. Minor groove in B form has a width 5.7 angstrom and depth 7.5 angstrom. In A form, it is wider. It is 11 angstrom and 2.8 angstrom. And in zDNA, it is 8.3 angstrom and 13.8 angstrom. What is the role of polymorphic structural changes in DNA, in its recognition? Because you know that the molecules, proteins, carcinogens, and many other molecules, they have to bind DNA and they recognize DNA base sequence. Now, the recognition is a chemical recognition and it depends upon the available functional groups for interaction to the ligand. Thus, as the DNA changes conformation from B to A and Z form and other forms, different functional groups get exposed to the solvent. Because of the difference in the groove sizes and changes in the surface area, the accessibility to a carcinogens, drugs, and protein molecule differs in this form. Latter leads to change in the interaction of DNA with the ligand and is of great importance for biological perspective. I want to give you a few examples of environmental dependent polymorphic transformations in DNA. For example, in sodium salt, 92% hydration, we get bDNA. In lithium salt, 66% hydration, we get bDNA. Or A DNA, we get it in sodium salt and 75% relative humidity. cDNA, we get it in lithium salt, 44% hydration. gDNA, poly-DG, poly-DC, and sodium salt, 2500 millimole, or magnesium salt, 700 millimole, or ethanol, 60%, or pyrimidine, the molecule gDNA is formed. Also, it can be formed in the presence of methyl cytosine, poly-DG, DMC, sodium salt 700 millimole, or ethanol 20%, spermidine 0.5%, we can get zDNA. I show you diagrammatically how DNA polymorphism is expressed. The figure is by Drew from Nature paper, and it shows how the change of salt concentration, the conformation B form can go to C form, or it can go to Z form can go to Z dash, A form can go to A dash, or B form can go to C dash. I wish to give you now few examples of sequence-dependent structural changes in DNA. In sodium salt, a synthetic DNA fiber poly-DA dt, poly-DA dt adopts a metastable A form and a stable D form, whereas poly-DG DC, poly-DG DC up to 92% relativity occurs in A form only. The spectacular BJ transition does not occur in poly-DA dt, poly-DA dt. I can give you few more examples of sequence-dependent structural changes in DNA. In the case of alternate AT sequence, long spline of water runs along the minor groove because of its lower electrostatic potential. For this reason, poly-DA, poly-DT does not undergo BA transition, and poly-DA dt, poly-DA dt has a metastable A form. Polynucleotides with guanosine replaced by ionosine, which lacks the N2 amino group, behaves as an AT polymer. Local distortion in DNA structure. In addition to the polymorphic changes, which are the structural changes of the whole of polymer, the DNA molecule, based upon the base sequence, has local distortions. This was first noted by Dickerson's group for a DNA dodecamer CG, CG, AA, TT, CG, CG. This DNA had overall parameters same as B structure, so it was bDNA, but there were local distortions in this structure leading to the bending of DNA by 19 degrees. To define these distortions, Drew's group took the axial system in DNA, that is, we show here in the picture view in the base plane. Like a dot here is the position of the helix axis, and we have dyad axis or a pseudo-dyad axis about which backbone has a dyad symmetry. Then we have a long axis which starts from C6 of pyrimidine to C8 of purine. So the three axes are helix axis, pseudo-dyad axis, and the long axis. And we define distortions with respect to these three axes. What are those distortions? Propeller twist, twist, tilt, and roll. Drew and Dickerson defined them as propeller twist is the total dihedral angle between the two hydrogen bonded base pairs. It is not connected to the helix axis system we discussed. The twist angle, generally given as TW, is the rotation angle between the successive base pairs about the helix axis. Tilt, theta t, is the rotation of base pair about dyad axis passing through the base plane. And roll, theta r, is rotation about an axis in the plane of the bases perpendicular to the pseudo-dyad axis. And these parameters for different base sequences are different, which define the local distortion in the DNA structure. This is shown more clearly in this picture, like we show here the propeller twist between the two base pair planes, twist angle, tilt angle theta t, and the roll angle theta r. Apart from double helical structures, there are DNA triplexes and DNA quadruplexes. What is a DNA triple helix? In a DNA triple helix, 3 DNA strands in helical symmetry bind over each other. The third strand, binds to a Watson-Crick base pair DNA through a hook chain, like a TA duplex can have a hook chain base pairing with poly TA, or reverse hook chain base pairing, like TA can have reverse hook chain with poly A. Homopyridine third strand is always parallel to the homopurine strand in pyrimidine purine X pyrimidine, whereas homopurine third strand is antiparallel to homopurine second strand in pyrimidine purine and third strand purine. We show in this picture hook chain and reverse hook chain base pairing in 2 triple helices. TAT, that is pyrimidine purine pyrimidine, and TAA, that is pyrimidine purine purine. In case of TAT, we show on the right hand side of the 2 bases adenine and thymine, and the left side thymine has a hook chain base pairing, that is the NH and O hydrogen bond with N7 of adenine and H N6 of adenine. On the right side, we show a reverse hook chain base pairing between 2 adenines rather, like N6H and N1, hydrogen bond with N7 and N6H of other adenine. We show in this picture a DNA triple helix, and the third DNA strand runs into the grooves of the hydrogen bonded Watson-Crick base pair DNA. There are various sequences, and there are many DNA sequences which can run as a third strand and which have great application in the field of molecular biology. Like DNA triplexes, we have DNA quadruplexes. In a quadruplex structure, 4 helically twisted DNA wind over each other to form a DNA tetrads. A typical example can be quoted, that of G4 quadruplex. Sequences rich in guanine are capable of forming 4-stranded structure with hook chain base pairing. The structures are stabilized in the presence of potassium ions. So students, what we have learned today? Like first, we have seen the historic Chargaff parity rules and their importance in polymorphic structural changes in DNA. Then we introduced you to the concept of DNA polymorphism, like variety of structures are formed under different environmental conditions, based on structures, based on sequences, and so on. Then we had discussed a left-handed zDNA structure, as it marked a very important transition in our understanding in DNA. I can tell you a joke. Rather, in the year 1980, when the left-handed DNA was discovered, everybody prior to that thought that all DNA has to be right-handed. And I was going in a cab in USA, and person asked me, "Where are you working?" I told I'm in cancer hospital. He said, "What are you working on?" I said, "DNA." He immediately asked, "Left or right?" I said, "How do you know?" You know, everybody talks about left and right-handed DNA. So it becomes such a popular issue. Newspaper, Economic Times, everybody started discussing, is DNA left or right, like two political parties. Okay, after that, now we elaborated on the structural differences between A, B, and Z form of DNA. Latter has great role to play in DNA recognition by enzymes, carcinogens, drugs, and metabolites. Again, I want to tell you a joke. I saw a cartoon, a lady which is totally clad in a dress where nothing except her little part of face and feet were visible. And the boy asked another, that, "Is she beautiful or not?" He said, "I see only feet and little part of face. I can't say." The same is the story of DNA. Like, the recognition is of the base sequence. You have to see the bases. And unless you see the bases, which you see only through the grooves, and the grooves are dependent on polymorphic structural changes in DNA, and they go on changing under different environmental conditions. So, the recognition of DNA by enzyme, carcinogens, and various proteins goes on changing depending on the environment. Then we saw that DNA does not have only long range distortion, but if you take a small segment, say dodecamer, CG, CG, AA, TT, CG, CG, this was given by Dickerson's group, and he described that this small sequence, based on exact sequence, there are distortion in the structures, and he defined them in terms of roll, theta r, twist, theta tw, tilt, theta, and propeller twist. These were explained on the basis of three axes, in the base, two in the base plane of DNA and one helical axis. Later, we introduced the reader to the basic concept in DNA multiplex structures like triplexes, quadruplexes, and so on. What we gained from it, the understanding of environment and sequence dependent structural changes, would now enable us to understand the intricacies in the regulation of protein synthesis by base sequence in DNA. Thank you very much for your kindness.